Good morning, we are team number 1, and we're going to talk about the sequencing of nucleic acids. Our team is conformed by me, Daniel, Elisa García, María Fernanda Uribe, and Claudia Zepeda. And first we'll start with the Maxam-Gilbert or chemical method, which is based on the sequencing of nucleotides by cleaves at specific sites. First, a radiolabeled single-stranded DNA is produced. Then, the bases in the DNA are chemically modified and removed with a specific base-dependent molecule. And subsequently, the phosphodiester backbone is cleaved using hot piperidine. And in the end of this process, as we can see in the picture, fragments that differ in length are produced. And these fragments can be separated using polyacrylamide gel electrophoresis. On the other hand, in the Sanger-Coulson method of sequencing, a copy of the DNA to be sequenced is made by the clenone fragments of the DNA polymerase. Firstly, a primer must be provided for the 3' terminus for DNA polymerase to begin synthesizing the copy. Subsequently, a radiolabeled DNTP is added into the DNA. These DNTPs lack a hydroxyl group at the 3' position of the deoxyribose backbone, impeding the chain elongation to progress. And finally, the deoxynucleoside triphosphates, or DDNTPs, are incorporated into the growing DNA intermittently. And this way, we produce a series of fragments at the specific nucleotides, similarly to the Maxam-Gilbert method.
Some of the applications of chemical sequencing can be to the oil, gas industry, and even the personalized medicine. Today, the Maxam and Gilbert sequencing is almost extinct because it's time-consuming, and using this method, you could only confirm about 200 and 300 bases of DNA every few days. Maxam and Gilbert sequencing also required working with large amounts of radioactive material and working closely to some neurotoxins. And also the simplification of Sanger sequencing caused chemical sequencing to lose its appeal. But this method led to the methylation interference assay, which is a major technique for studying the interaction of proteins with major groove guanines, adenines in the minor groove, and cytosines in single-stranded DNA. And this method is used to map DNA binding sites for DNA binding proteins. And this resulted in the relatively rapid analysis of contact sites for DNA binding proteins.
The enzymatical sequencing method has given rise to many new technologies. One that is very important is known as next generation sequencing. This has been a major scientific breakthrough because it has a lower cost and it has a high throughput sequencing. So this means that it allows millions of reactions simultaneously to occur. And here in the image, there are the main steps. So first, in the library preparation, the fragments of DNA are tagged with adapters. And then each fragment is amplified and the bridge amplification takes place. And then they're joined in clusters of similar groups. And then this is done to be able to perform the library sequencing. And the library sequencing is done by sequencing by synthesis, which what happens is that the elongation of the primer is the first thing to happen. And then the primer is elongated with the different bases, and these bases have a fluorescent reactive, which is what emits the signal and is what allows the visualization of the sequence to be performed. And this is based on the Sanger method. It was what gave way to this new method. And even though these new methods are very useful and cost effective, the Sanger method is still being used nowadays in different scientific researches. In the next slide. the Sanger method was used in immunoglobulin heavy chain variable gene alleles detection. So in this paper, the researchers wanted to identify new alleles. And in order to do so, they did use new next generation sequencing. However, they still use the Sanger method in order to validate these new alleles. And it is very useful because it also allows a more specific identification of the genes. And in some cases, it is easier to use the Sanger method because it can sequence smaller strands of DNA, and it doesn't have to be necessarily a next generation sequencing with all of the millions of copies. So in this paper, they still used the method of Sanger combined with the next generation sequencing. So these two combined can be of great use for the medical field and in the use of vaccine development or cancer treatments.
The chemical and the enzymatic methods set out the basis for development of laboratory equipment to make the process of sequencing more efficient. Sanger sequencing is the most used method. One example of this is this equipment, a spectrum compound CE, that is used for Sanger sequencing and fragment analysis. Also, is used to confirm successful genome edits in transform culture. Although the chemical method also was a very important discovery, it's no longer used widespread because of the radioisotopes. So instead, the next generation sequencing methods are used to develop laboratory equipments that are capable of sequencing nucleic acids. One example is this equipment that is called MySeq system, and it's used the sequencing by system synthesis SBS as a principle, and is capable to deliver 600 bases of sequence data per read.
And here are our references, and thank you.